Thank you for giving me this opportunity. I will be talking about anti-parasite activity of edible mushroom, the Morel edible mushroom. I am sure many of you know it. So as I said, I'm working again, just I will start again. I'm working in the MIGAL-Galilee Research Center, which is focusing on environmental studies and food sciences, agriculture a lot, and also Tel Hai College, also in Northern Israel. The work was done also in collaboration with the Baruch Padeh Medical Center in Korea. I'm talking about leishmaniasis. Leishmaniasis is a unicellular parasite with this flagellum. It's infecting a lot of in different ways. There are several symptoms of, for example, the skin symptoms. There are also another way of infection is skin, mouth, and nose. And another way of infection is beside the skin is also inside the person, which means that there is high fever, low red blood cells, and enlarged spleen and liver. And there are different ways that people are, the symptoms are different in different people. The problem is that it is very, very worldwide infection. The distribution is all over the world. A survey in 2016 showed that there is at least 23 species of this parasite over the world. Over 88 countries are infected. We can find the parasite in over 88 countries. Around 350 million people are at risk. Of course, many of them are in the poor countries. 12 million are infected, and every year around 1 million of people are newly infected with this parasite. The thing is that also the household animal, like dogs and horse, may be infected, and this is increasing phenomenon now. The life cycle of the parasite is very complicated. It's actually a combination of 3 cycles. The first cycle is in the host, in the vector, in the reservoir animal, for example, the dog, where the parasite is, of course, infecting the dog, entering the macrophages, where in the macrophages, the parasite loses its flagellum, and it becomes inside parasite. I mean, it changed from mastigote phenotype to a mastigote phenotype, which is without the flagellum. It is multiplied in the macrophage, and after a while, the macrophage will explode, and all the parasite will go outside into the bloodstream. The other cycle is the sand fly. The sand fly can bite the dog and take into its blood the cells of the parasite, and the parasite there then it can bite, of course, the people, and transfer the parasite into people. And also in the people, there are another cycle where the parasite with the flagellum becomes again losing its flagellum and become a mastigote phenotype, multiplying the macrophages, and then when the macrophages is deteriorated, these cells are going to the blood, and again all these cycles going over and over and over again, which means again that we need animal to be the reservoir for this parasite. In Israel, there is increasing cases of this leishmania in recent years. Also over the world, there is a problem of the changing in climate change problem that might be the cause of increased cases of this leishmaniasis. If we look here, there is increase of, in Israel at least, there is increase of 300% in the infected cases from the 19th until 2017. We have two major species of the Leishmania, which is Leishmania major and Leishmania tropica. It's spread over Israel, one in the north and the other in the south of Israel. And the reservoir animal is Procavia, which is protected animal in Israel. It's its nature, it is in the nature, it's not household animal. It found everywhere, but it is protected one. So we have a problem of the reservoir itself. So what we can do? The treatment nowadays are, we have several drugs, local or oral drugs like ketoconazole and others. And in severe cases, we use the amphotericin B. But the problem with all these drugs is that they need prolonged treatment. They have toxicity of the people, also to the human. I mean, it might cause side effect. And another problem is with the use of these drugs, there is resistance in the Leishmania cases, in the Leishmania parasite. And all this show us that we need another way to treat the Leishmania and to find alternative ways. I'm a mycologist, so I work with edible mushrooms, and we know that there is increasing awareness about the use of edible mushroom or medicinal mushroom in every aspect of health. They have a lot of health benefits like anti-tumor, antioxidant, antibacterial, and many other activities. There is I'm sure you know, Ganoderma lucidum, Pleurotus striatus, Trametes versicolor, Lenticinus odidus, Cordyceps, and Agaricus. Well, all of them are medicinal mushrooms that are actually used in the traditional medicine. But we were looking about anti-parasite activity, and actually there are several cases that were also studied. All this medicine aspect in medicinal mushroom, also anti-parasite activity, with also specifically working on Leishmania, extract from many mushrooms was found to be to have also anti-Leishmania activity, and even there was also isolation of specific terpene that was used also to treat Leishmania very successfully, but it was also only in an experimental way. I'm working a lot with the Morel mushrooms, and the Morel mushroom is also medicinal mushroom. It has antioxidant, antibacterial, anti-tumor, anti-inflammatory. Many variability of beneficial health benefit of this mushroom. There are different species of the mushroom. I'm sure you know them. And we were using them as a source for anti-parasite activity. So what we did is extract fractionation of water, ethanol, polysaccharide fraction, and polyphenolic fraction. And, sorry, can you mute it? Because, okay. And we used it to study the in vitro inhibition of Leishmania. And we did also a recovery test. So what we saw that there is all the fractions that we used, all of them had anti-Leishmania activity. The water fraction, the ethanol fraction, the water-soluble polysaccharide fraction, and the polyphenolic fraction, all of them had anti-Leishmania activity. And as we see it here, with the water fraction and the ethanol fraction being the most active ones. When we look at the time course of the activity, we see that with time there is reducing in the number of the live leishmania, as we see it here. Again, all the fractions were active, where some of them were more active, the other were less active, but in any case, all of them with time, the leishmania cells, the live leishmania cells decreased. We look at the microscopic observation as we see it here. We see the pomastigot cells of the leishmania with the flagellum. But when we add the extract, there is aggregation of the leishmania and then also totally deterioration of all the cells. This is in the observation of the microscope. Okay. Another thing that we do in order to see if there is any recovery, I mean, if we are taking the extract out of the system, what will happen then? Are the cells able to recover from the problem of the extract? And as we see here, we added several concentration of the extract, and this is the number of before the recovery cell. This is after the recovery cell, and we see that there is no recovery of the cell. All of them continue to decrease in the number of the live cell and no recovery of the leishmania cells. So we took it also further and we examined it also in all the work that I have shown you was on leishmania measure, but we did it also with the Tropica. We got the same phenomena that with the extract, there is decreasing in the live cells, where in the control, there is a steady number of the live cell. With the time, there is aggregation, but no deterioration of the cell. This is the same with leishmania major and leishmania tropica. And as a summary, Morchella extract showed anti-leishmania activity against both tropica and major. All extract fraction has positive activity with the waters and the ethanol were the most active ones. Of course, further study, we are conducting a further study to study also the, this was only on the pomastigot stage of the leishmania. We are going to study it also on the amastigot phenotype of the leishmania, and then to try to use it as a treatment. Thank you very much.